Assalamu alaikum everyone. I am Dr. Muneeba Sayeed, and this is the lecture 4 of AMR series. And it's about antibiotics, the classification and spectrum of activity. So we've got different lists. This is the list of antibiotics that we have, which includes penicillins, cephalosporins, carbapenems, beta-lactamase inhibitors, quinolones, glycopeptides, aminoglycosides, and many other antibiotics. And before prescribing antibiotic, if you look at this list, so it's a very tough choice to make actually. So before prescribing any antibiotic, it's very important to have the background knowledge about the class to which this antibiotic belongs and what are the bacteria does it cover. Antibiotic classification. Basically, antibiotics are classified in three ways. Bactericidal or bacteriostatic antibiotic based on mechanism of action and based on the spectrum of activity. So bactericidal antibiotics are those which actually kills the bacteria directly, while bacteriostatics are ones which prevent them from dividing. So we have a list of bactericidal antibiotics, which includes penicillins, cephalosporins, aminoglycosides, quinolones, vancomycin, trimethoprim, sulfamethoxazole combination, metronidazole, polymyxins, bactericin, and nitrofurantoin. While bacteriostatic antibiotics includes tetracyclines, macrolides, chloramphenicol, lincosamide, linezolid, sulfa drugs, and trimethoprim. Bacteriostatic antibiotics, since they prevent the bacteria from dividing, so you cannot give them in bacteremic situation, except two antibiotics, which includes macrolides, that is azithromycin, which is given in Salmonella typhi bacteremia, that is enteric fever, while linezolid is given in case of vancomycin resistant enterococcus bacteremia, while rest of the bacteriostatic antibiotics are not given in bacteremic situation. The other way of classifying antibiotics is actually based on the mechanism of action. So based on mechanism of action, we need to know the actual structure of the bacteria. Bacteria has got a cell wall, then there is a plasma or cell membrane. It has got cytoplasm, then ribosomes and chromosomes. So the antibiotics that inhibit cell wall synthesis are called the cell wall synthesis inhibitors. Then we've got protein synthesis inhibitors. Then we have nucleic acid synthesis inhibitors, folic acid synthesis inhibitors, and cytoplasmic membrane inhibitors. Beta-lactam antibiotics, beta-lactamase inhibitors, vancomycin, polymyxin, and bactericin, these all belong to cell wall synthesis inhibitors. While protein synthesis inhibitors include 30S subunit inhibitor and 50S subunit inhibitor. Nucleic acid synthesis inhibitors include quinolones, metronidazole, and nitrofurantoin, while sulfonamide and trimethoprim comes under the classification of folic acid synthesis inhibitors, and colistin and polymyxins, these are the cytoplasmic membrane inhibitors. Starting from cell wall synthesis inhibitors and beta-lactam antibiotics, very important class of antibiotic. And these are called beta-lactam antibiotics because they've got a beta-lactam ring in their molecular structure. And these antibiotics include penicillins, cephalosporins, monobactams, and carbapenems. And all these antibiotics work by inhibiting cell wall synthesis. Starting from penicillin, so penicillin basically further divided into natural penicillins, which includes penicillin G, available in IV form, and penicillin B, which is available in oral form. We've got aminopenicillins, ampicillin, available in IV form, while amoxicillin in oral form. Then we have anti-staph penicillin, which is also known as the penicillinase resistant penicillin, and it includes methicillin, oxacillin, neficillin, and dicloxacillin. Only diclox is available in oral form. Then we have anti-pseudomonal penicillin, which has activity against the pseudomonas, and it's piperacillin and ticarcillin. So starting from aminopenicillins are the drug of choice for ampicillin-sensitive enterococcus, while anti-staph penicillin covers group A streps. The anti-pseudomonal penicillins, that is piperacillin and ticarcillin, they are divided into two groups, that is uridopenicillins and carboxypenicillins. Moving on to cephalosporins and cephamycins. So cephalosporins, these are the different generations of cephalosporins. One thing to remember here is that as you ascend up in the generation, moving from first generation to fourth generation, there is more gram-negative coverage compared to gram-positive. So basically, we have got two types of bacteria, as you all know, gram-positives like Streps, staph, these are, or enterococcus, these are the gram-positive bacteria. While gram-negative bacteria includes Enterobacteriaceae, Pseudomonas, Acinetobacter, etc. So as you go up to from first generation towards fourth generation, there is more gram-negative coverage and lesser gram-positive coverage. So first generation cephalosporin includes cephalexin, which is available in oral form, cefazolin, excellent drug which has MSSA, that is methicillin sensitive Staph aureus activity, and cefazolin can be easily given in cases of MSSA bacteremia. And we've got cephradine, which is available both in IV and oral form. So first generation, more gram-positive coverage. Second generation includes cefaclor and cefuroxime. Then we've got cefamycin. Cefamycin is the only group of the cephalosporin which has anaerobic coverage, includes cefotetan and cefoxitin. Third generation cephalosporins are very commonly used. The per oral form is cefixime and cefpodoxime. So cefixime, what's the difference between cefixime and cefpodoxime? So cefixime has more gram-negative coverage. So if you are giving it for any abdominal infection, you would go for cefixime, while cefpodoxime has more gram-positive coverage. Like in cases of strep pneumo, you can go for cefpodoxime. While IV forms are ceftriaxone, cefotaxime, ceftazidime, and cefoperazone. Then we've got fourth generation cephalosporins, which are the cefepime and cefperome. And we've got MRSA active cephalosporins, which are the ceftaroline and ceftobiprole. MRSA active cephalosporins are not available in Pakistan. So as I've mentioned earlier, first generation has more gram-positive coverage. Second generation maintains gram-positive activity, but also has increased gram-negative coverage. Cefamycin's only anaerobic coverage. Third generation, like ceftriaxone and cefotaximes and ceftazidime, they all have good CNS penetration. That's why they're used in case of meningitis. Cefotaxime and ceftriaxone has pneumococcal coverage. So it's given for both streptococcus associated pneumonia as well as in meningitis. Ceftazidime has pseudomonal coverage. So you need to remember this, that what are the which antibiotics in the cephalosporin group has pseudomonal coverage. So if you are going to treat pseudomonas infection, choose ceftazidime, not ceftriaxone. Fourth generation, there is no oral form. It includes cefepime. Again, it has very good CSF penetration. It has the widest spectrum of all cephalosporins. It covers pseudomonas, it covers enterobacter, MSSA, pneumococcus, strep, and that's why it's a drug of choice for febrile neutropenia. MRSA active cephalosporins, their gram-negative activity is same as that of third generation, but they also have coverage for MRSA, that is ceftaroline, covers MRSA, pneumococcus, and enterococcus faecalis, while ceftobiprole covers MRSA-resistant pneumococcus and pseudomonas. Moving on to monobactams. So this is a class of antibiotic, which is again a beta-lactam antibiotic. It has pure gram-negative coverage, no anaerobic or atypical coverage, no gram-positive coverage. It has anti-pseudomonal activity, no major toxicities is seen, can be used in case of penicillin allergy, usually given in cystitis, pyelonephritis, low respiratory infections, wounds, diabetic foot, but you need to add gram-positive and anaerobic coverage if you are using Astronam, not readily available in Pakistan. Moving on to carbapenems. So carbapenems, this includes ertapenem, imipenem, meropenem, and doripenem. Ertapenem, once daily dose, available in IV form, but it's expensive. It is easily hydrolyzed, so can be used to detect carbapenem resistant enterobacterials once daily dosing is required. If we compare imipenem and meropenem, their coverage is more or less same, but imipenem has more gram-negative coverage, while meropenem has more, imipenem has more gram-positive coverage, while meropenem has more gram-negative coverage. And DORI has very good activity against Pseudomonas aeruginosa. So carbapenems, gram-positive, gram-negative anaerobic coverage, no MRSA coverage. Stenotrophomonas and Burkholderia sebaceae are inherently resistant to carbapenems, a point which you need to remember. There is no cross sensitivity, another very important point. Like, for instance, if you see a culture report in which you see that the organism is sensitive to meropenem, it certainly does not mean that this organism is also sensitive to imipenem. You need to call the lab and ask them to check whether it's sensitive to imipenem or not. Same is the case. Same is the case with ertapenem. If the organism is sensitive to meropenem, it certainly does not mean that it is also sensitive to ertapenem. You need to check it also. Meropenem, better pseudomonal coverage than imipenem, and ertapenem is not active for Pseudomonas and Acinetobacter, so cannot be used in ventilator-associated pneumonias. So since carbapenems have anaerobic coverage, so there is no need of giving metronidazole along with carbapenems to treat abdominal infection, a point to remember. Then moving on to beta-lactamase inhibitors. So we've got, we know that the beta-lactam antibiotics are called beta-lactam because they've got a beta-lactam ring inside. When they act on the susceptible bacteria, the bacteria dies. When the beta-lactam antibiotic acts on a bacteria which produces beta-lactamase, the antibiotic ring would be destroyed and the bacteria survives. The beta-lactamase inhibitors, adding beta-lactamase inhibitors in the antibiotic would act against those bacteria which produces beta-lactamase and the bacteria dies. So this is given in combination with beta-lactams. These are called the suicidal drugs and they have anaerobic coverage. It includes clavulanate like amoxicillin. It's a beta-lactam antibiotic. It's a penicillin. When you combine it with clavulanate, it becomes a beta-lactamase inhibitor. Sulbactam given in combination with cefoperazone. Tazobactam is given in combination with piperacillin. Avibactam, which is given in combination with aztreonam. So of these, avibactam aztreonam combination is not available, while all above three are available in Pakistan. Now moving on to vancomycin. So vancomycin is a glycopeptide. It inhibits the latest stage of bacterial cell wall synthesis. It is active against gram-positive, all the gram-positives. There is no activity against gram-negative. It is nephrotoxic and you need to monitor the trough levels. Trough levels are the levels which are drawn 5 minutes before the next dose of antibiotic. Usually trough levels are checked before 4th dose of antibiotic. And the level should be between 15 to 20. Polymyxins. So polymyxin, it penetrates the outer cell membrane of the bacteria and disrupts its bacterial cell membrane. It has activity against gram-negative bacilli. So we give it for MDR Acinetobacter or MDR Pseudomonas aeruginosa. It has poor activity against Proteus, Providentia, Burkholderia, Brucella, Serratia, Aeromonas, Moraxella, Morganella, Edwardsella. Most anaerobes are resistant to polymyxins. It is bactericidal, but it does not go well into the lung parenchyma. So we may need to add colistin nabs in case of refractory health HAP or ventilator-associated pneumonia. So now the next class of antibiotics is the protein synthesis inhibitors. Starting from aminoglycosides, so the list of aminoglycosides includes amikacin, streptomycin, gentamicin, tobramycin, and neomycin. It binds to 30th subunit of our ribosome. It is concentration dependent. You need to give a larger amount of antibiotic to reach a certain concentration that would cause the killing of the bacteria. So single dose would be enough. Post-antibiotic effect is there, that even after the last dose, the antibiotic keeps on circulating into the system and its effect would last for more than the usual time. Synergism, you give it with other antibiotics to potentiate the effect of other antibiotics, like in case of endocarditis. It is bactericidal, but it will not work in acidic medium like abscess. So no point of giving amikacin in case of lung abscess or abscess anywhere in the body. It has antimicrobacterial activity and is also used in case of tuberculosis. You may go for multiple dosing or single dosing. Then tetracyclines. Tetracyclines enter the bacteria through porin channels. It inhibits protein synthesis. There are three types of tetracyclines. Short-acting includes tetracycline peroral. Intermediate acting includes demiclocycline, and long-acting include doxycycline, minocycline, and tigecycline. Doxy and minocycline are available in oral form, while tige is available in IV. It has activity against different organisms like anthrax, Bartonella, Brucella, Chlamydia, Campylobacter, Clostridium, Leptospira, Mycobacterium, Nocardia, Rickettsia, also given in case of prophylaxis against anthrax, malaria, and plague. Then the macrolides. So macrolides are the erythromycins, clarithromycin, and azithromycin. Now here you need to remember the difference. Clarithromycin has more gram-positive coverage, while azithromycin has more gram-negative coverage. That's why azithromycin is given for typhoid, while clarithromycin has been given for community-acquired pneumonias. Macrolides inhibit protein synthesis. Clarithro and azithro can be given for community-acquired pneumonia, pertussis, Mycobacterium avium complex infection, and Lyme's disease, Babesia, malaria, H. pylori, Campylobacter, and Cryptosporidia. Single azithromycin dose is given for sexually transmitted diseases and trachoma. Clindamycin, again a protein synthesis inhibitor, and it's active against pneumococcus and group A straps, but resistance is increasing. It also acts against MSSA and methicillin-resistant Staph aureus. Most anaerobes like Bacteroides fragilis are sensitive to it, but resistance is now seen. Also, it acts against Clostridium species. It is being used for Pneumocystis jirovecii, toxoplasmosis, and malaria. And all enterobacterials are resistant to it. So clindamycin has gram-positive coverage and it has anaerobic coverage. It has no gram-negative coverage. So you cannot use clindamycin for E. coli or Klebsiella. Linezolid, again, a protein synthesis inhibitor. It has activity against gram-positives like MSSA, MRSA, Enterococcus, Foraini, Listeria, Bacillus. It's used in vancomycin-resistant Enterococcus. It is used in MRSA pneumonia because it has excellent lung penetration. Skin and soft tissue infections, ventriculitis, meningitis, endocarditis, but it should not be used in case of MRSA bacteremia because it is bactericidal. Also, one should not readily use linezolid. Linezolid is a controlled drug, and it should be a controlled drug in pharmacy, and only ID specialists should prescribe this in antibiotics because random use of linezolid will lead to resistance, and we need linezolid for hospital-acquired resistant bacteria like vancomycin-resistant Enterococcus. Then we have got nucleic acid synthesis inhibitors, and these are the quinolones. We've got first generation, second generation, third generation, and fourth generation quinolones. Nalidixic acid and oxalinic acid are the first generation, and then fluorine was added, so they become oxofloxacin, norfloxacin, ciprofloxacin in second generation. Third generation includes levofloxacin, and fourth generation include moxi and gatifloxacin. All three, that is levo, moxi, and gati, are actually the respiratory quinolones. So they inhibit the enzymatic activities of DNA gyrase and topoisomerase and promotes cleavage of bacterial DNA. Nalidixic acid is the first quinolone, and then fluorine was added to form ciprofloxacin, which has more potent gram-negative coverage. Anti-pseudomonal quinolones include levofloxacin and ciprofloxacin. So you need to know if you are looking at Pseudomonas, prescribe either Cipro or levofloxacin. Then we've got respiratory quinolones, which includes levofloxacin, gatifloxacin, moxi, and gemifloxacin. They have pneumococcal coverage. So in case of pneumonia, only prescribe these antibiotics if you have to prescribe one. Agents of atypical pneumonia, you can also use it. And amongst all these, only moxifloxacin has anaerobic coverage. Then we've got the folic acid synthesis inhibitors. These inhibit folic acid synthesis, like sulfonamides inhibit bacterial growth by interfering with folic acid synthesis, and trimethoprim blocks the next enzyme step in folic acid synthesis by inhibiting the dihydrofolate reductase. It's active against nocardia, stenotrophomonas. As I've told earlier, that stenotrophomonas is a bacteria which is inherently resistant to carbapenem. So you can use this combination in those cases: atypical mycobacterium, meningitis, PCP, and toxoplasmosis. Then we have MRSA, skin and soft tissue infections, and it is also used in case of UTI. Tigecycline, it has no anti-pseudomonal coverage. It is bacteriostatic. And this is the last resort, actually. It is not a very good drug because it's bacteriostatic, used in caution in cases of bacteremia and healthcare-associated pneumonia. So now that we know that the different classes of antibiotic, it's time to look at the spectrum of antibiotic. Which organisms do they cover? So this is a list of organisms. The purple ones are the gram-positive organisms, while the red ones are the gram-negative organism. Starting from the purple one, the gram-positive, we have Streptococcus, we have methicillin-sensitive Staph aureus, we have Enterococcus M-sensitive, methicillin-resistant Staph aureus, and vancomycin-resistant Enterococcus. While moving on to the red side, we have enterobacteriales like E. coli, Klebsiella, Pseudomonas aeruginosa. Extended spectrum beta-lactamase producers. ESBLs are those which are resistant to ceftriaxone, while CRE is the carbapenem resistant enterobacteriales, which are resistant to meropenem, imipenem, and multi-drug resistance are those gram-negative organisms which are resistant to 3 or more class of antibiotics. So amoxicillin, ampicillin, it has gram-positive coverage and it covers some of the sensitive E. coli and Klebsiella. Anti-staph penicillins, neficillin, oxacillin covers only strep's and staph. As I told you earlier, that first generation and first generation cephalosporins has more gram-positive coverage, some gram-negative coverage. Ceftriaxone, cefotaxime, again positive and some gram-negative coverage. From ceftazidime and cefepime, they have pure gram negative coverage. Cefepime also has gram-positive coverage. It covers MSS and streps. Then we have carbapenems. Carbapenems covers has gram-positive as well as gram-negative coverage. It covers ESBL2. Same is the case with piperacillin tazobactam and cefoperazone sulbactam. Vancomycin, purely gram-positive, no gram-negative coverage. Linezolid, again gram-positive, and it covers vancomycin-resistant Enterococcus 2. Quinolones, ciprofloxacin, it covers only gram-negatives. Cipro should not be used for MSSA infection. Then moxi and levo, it covers strep, MSSA, and enterobacteriales. Amikacin, it has a wide spectrum, but it's not used much because of the nephrotoxicity it causes. And also then we have trimethoprim sulfamethamazole. It again covers gram-positives. It has no pseudomonal coverage. And tigecycline is given. It has got a very broad spectrum, but it should not be used readily. We need to keep it, reserve this antibiotic for only largely resistant organisms. And colistin, again, gram-negative coverage, and it covers the multi-drug resistant organism. So, and if you look at the gram-negative anaerobes, these are being covered by clavulanate, metronidazole, carbapenem, piperacillin, tazobactam, and moxifloxacin. So, if you are prescribing piperacillin, tazobactam, or any carbapenem, you don't need to give flagell along with it. These alone have anaerobic coverage. No point of adding metronidazole along with carbapenems or piperacillin, tazobactam. So now with this background knowledge, is it a really tough choice? It is not a really tough choice if you know which disease are you treating, what is the site of infection, which which bug you are targeting, and then you can easily make out which drug you need to give. So if you are seeing community-acquired infections like ampicillin-sensitive enterococcus, streps, or enterobacterials like E. coli or Klebsiella, anaerobes, you can easily give cephalosporin and ceftriaxone, ceftazidime, cefepime, and for anaerobes, you can give metronidazole, clavulanate, ciprofloxacin, moxilevofloxacin. And if you are dealing with hospital-acquired infection where you are suspecting vancomycin-resistant enterococcus, MRSA, Pseudomonas, ESPL, or CRE or MDR, you can go for piperacillin, tazobactam, cefoperazone, sulbactam, carbapenems. For MRSA, vancomycin, linezolid, amikacin, tigecycline, and colistin. So which drugs have anti-MRSA activity? Vancomycin, clindamycin, trimethoprim, sulfamethamazole, doxycycline, linezolid, ceftaroline, ceftobiprole, and daptomycin, they all have anti-MRSA activity. Piperacillin, tazobactam, carbapenems, cefepime, ceftazidime, ceftobiprole, ciprofloxacin, aminoglycosides, polymyxin, estrogen, they all are anti-pseudomonal. For the gram-negative anaerobes, we have got, and positive ones, some of the positive ones, we have beta-lactam, beta-lactamase inhibitors, we have cefamycin, metronidazole, carbapenem, moxifloxacin, clindamycin. These all have anaerobic coverage. Like, for instance, if you're dealing with aspiration pneumonia, so moxi would be enough because it is a respiratory quinolone and it would also act on the anaerobes. So alone moxi would be enough for treating aspiration pneumonia. For carbapenem resistant enterobacterials or multi-drug resistant organism, you have the choice of colistin, tigi, and amikacin. Also fosfomycin is used. So remember, never abuse these antibiotics. First, see which disease the patient is suffering from, what is the likely bug, which antibiotic you need to give. Do you need a antibiotic with a gram-positive coverage or an antibiotic with a gram-negative coverage, and then prescribe it accordingly. And save our precious antibiotics. Don't misuse the antibiotics. So with this, I end here. Thank you so much. And if you have any question, you can write it in the comment section.